Very good morning to the attendees of breakfast at UMHealth. So today we have another set of interesting talks. Let me start off with the talk by Dr. Kimberly Fornes. So Dr. Kimberly Fornes is a special epidemiologist focused on emerging zoonotic and vector-borne diseases. Kimberly's research focuses on the impact of environmental change on infectious diseases dynamics. She uses a range of health observation data, geostatistical model methods, and machine learning approaches to monitor environmental changes and assess the impacts on infectious disease transmission. Based in Malaysian Borneo for over 6 years, a core focus of this work has been characterizing the spatial epidemiology of emerging zoonotic malaria in Malaysia and the Philippines and evaluating association with deforestation and other land use changes. Her current research program is funded by the Wellcome Trust and Royal Society and aims to identify how environmental data can be integrated into infectious disease surveillance system. Dr. Kimberly, the screen is yours.
Excellent. Well, thanks so much for the introduction and for having me here today. So I'll just share my slides. Okay, I think those should be up now. Great. Okay, well, I'm going to speak a bit today about how we have used Earth observation data to actually understand the emergence of zoonotic malaria in Malaysia. So this is actually work that we've been doing in collaboration with the University of Malaya for now since 2013, so over 10 years now. So it's been a very long-standing research collaboration. So just for those of you who aren't familiar with Plasmodium knowlesi, this is the malaria parasite known as monkey malaria or Malaria monyet. So this parasite is typically carried by long-tailed and pig-tailed macaques, and it can be carried by other non-human primates. Like all malaria species, this is transmitted by Anopheles mosquitoes, so it's believed most of the transmission occurs when an Anopheles mosquito bites an infected macaque and then goes on to bite a person. But of course, one of the reasons we're really quite concerned about Plasmodium knowlesi is this can cause severe and fatal malaria in people. And really in Malaysia, we've seen something quite remarkable in terms of the trends. So while Malaysia has a really highly effective malaria elimination program, I would say one of the best in the world, and this has really brought down malaria cases across Malaysia in the past 2 decades, we've seen this corresponding increase in the number of zoonotic malaria cases. So Plasmodium knowlesi is now the only indigenous cause of human malaria in Malaysia and has been since 2018. And this causes thousands of human cases every year. So one thing to really highlight is that this is not just a Malaysian problem. Plasmodium knowlesi causes human malaria cases across Southeast Asia and parts of South Asia. But really, there's a substantial spatial heterogeneities in this. So there are areas where we see much higher numbers of cases, particularly in Malaysian Borneo. And really, the epicenter of where we see the highest number of cases is in Sabah, in Malaysian Borneo. So kind of when we first started working on this over 10 years ago now, we started kind of thinking about why would we see such a pronounced increase in human Plasmodium knowlesi cases within these particular regions. And one thing that was really quite immediately clear when we looked at a map of deforestation during the same time period is that during this time, these 2 decades where we really saw the emergence of Plasmodium knowlesi, these areas that were reporting some of the highest numbers of human Plasmodium knowlesi cases also had some of the highest rates of deforestation globally. So kind of one of the first things we wanted to look at is there an association between deforestation and increased human risk of Plasmodium knowlesi? So the first study we looked at was actually in Kudat and Kota Marudu in northern Sabah. We did a retrospective review where we looked at hospital-based reports. We assembled all the data on malaria cases. We geolocated these down to kampung level, and we fit statistical models to adjust for some of the uncertainties in diagnosis during this time period. And what we found when we overlaid this data with a number of different environmental variables, so things like vegetation, elevation, and forest loss, is we found that there was actually, in fact, very strong associations between village level incidence of Plasmodium knowlesi and both forest cover and forest loss. So really the places where we were seeing the highest number of human cases of Plasmodium knowlesi coming from had these quite patchy landscapes. So areas where we have some forest remaining, but also forest being cleared. And so really this raises the question, why? So why do we see this association, and what are some of the mechanisms that are underlying this type of risk? And so of course, kind of just thinking about why we would see an increase in zoonotic malaria in areas that are being deforested, we know that land use change and other environmental changes can have impacts on a number of different components that relate to infectious disease transmission. So for example, we know, of course, that forest loss will impact wildlife populations. So how many macaques are present in an area, where they're moving to, what types of food they're eating, how close they are to people. Of course, as well, when we have these kind of large-scale land clearings, this impacts vector ecology. So this can create new breeding sites for different types of mosquitoes. This can favor different mosquito species and can also change the behavior of these mosquito species or their preference for biting different animals versus people. And then, of course, kind of one of the main things when we see an area that's being cleared or these kind of extensive developments is generally this impacts us and our movements. So someone is probably doing the clearing. Often when we see these areas, there might be logging camps moving into a new area. So this could increase the population of susceptible individuals within a particular area. And of course, this impacts their access to different control measures and health systems. So often we find that some of these populations that have the highest risk are living in some of the most remote areas. And these have some of the lowest access to healthcare. So the hospitals and clinics can be quite far away and difficult to get to. So kind of a core component of actually trying to understand these different dynamics is actually thinking about not just how do we monitor these different populations, but how do we actually monitor land use change and other environmental factors. And really the main way that we do this is using Earth observation data. So Earth observation data is exactly like what it sounds. This is any type of data we can use to observe processes that are occurring on the Earth's surface. So I'm sure everyone is using Google Maps and Google Satellite and is very familiar with satellite images. We use a wide number of other types of satellite-based data sources. So these have actually been around for quite a long time, since the 1970s. But we've really seen an increase in the quantity and quality of satellite data that we can use to monitor some of these changes. We've also increasingly been using small low-cost drones, so we can do some very targeted mapping. So to look in specific areas and collect very fine-scale maps of different landscapes, so we can monitor changes as they're happening. So as an example of some of the different types of things that we can observe from Earth observation data, we of course can't see the parasite, but we can characterize a number of different factors that are related to malaria transmission. The first is thinking about landscape composition. So we can look at some of these much wider factors that are associated with forests and forest loss. So what types of trees are in a particular area? How quickly is it changing? We can also, in some cases, look at different types of aquatic habitats that may be suitable for different mosquito vectors. And this is a bit different from Malaysia, but often we can find in Africa, we find that certain malaria vectors are breeding in some of these large open water bodies, and we're able to actually directly observe those through Earth observation data. And finally, one of the things we often forget is that we can use different types of Earth observation data to actually gather quite good data on human and wildlife post distribution. So we can monitor things like houses, the development of informal settlements or roads, and in some cases even directly observe animals using drone surveys. So just some examples of what some of this data looks like and some of the different types of information we can obtain from this. This is an example using the Landsat satellite data. This is a satellite that's run by NASA, which is freely available to everyone in the world. And what this does is this collects a lot of images of the Earth's surface, and then we can use machine learning models to actually fit different approaches to classify these patterns of land use change. So some examples that I've shown here, this is an example of looking at small-scale agriculture, where we can actually pick up these small-scale clearings, which are associated with different agricultural practices. We can, of course, kind of monitor some of these larger-scale clearings associated with plantations, looking at things like roads or other settlements that might, developments that might impact risk. So this is an example of gold mining in South America here. We also increasingly have been using drones. So we're able to collect a lot of very detailed imagery from using drones of kind of fine scale landscape changes. And really one of the things that's changed very rapidly within the past year or two is the development of some of these artificial intelligence approaches, so deep learning that we can use to rapidly analyze these images and pick out some of the features that we're interested in. So this is actually an example from West Africa, but looking at how we can pick out different types of vegetation or water bodies that might be associated with risk and automatically detect these and classify these from drone images. And of course, kind of the purpose of collecting all of this environmental data is really not just to monitor the environment, it's to think about how the environment is impacting all of these different populations that influence zoonotic malaria risk. So one of the main questions when we're looking at Plasmodium knowlesi is actually where are the macaques and other non-human primates moving and roosting, and how can we actually characterize where they are relative to the environment. So this is actually an example using a GPS collar to collect data on where the particular macaque troop is moving along the edge of a plantation and the border of a forest. And then also looking at where they're roosting at night. So where are they when the mosquitoes are most likely to be biting them, and how close is this to human settlements? We also, of course, do a lot of work, particularly with the University of Malaya, mapping mosquito habitats. So looking across kind of wide spatial areas, how can we kind of understand the distribution of the Anopheles mosquito vectors and how this varies by habitat type. So this is some examples again from northern Sabah, but looking across different types of forest or agriculture and quite close to kampongs. Collecting a lot of data using human landing catches. So actually counting the number of mosquitoes that are coming to bite a person within a given night. And also we've increasingly been looking at how we can catch mosquitoes that are higher on the canopy that may be biting macaques. So this is something we're still trying to develop. But from this data, we get a lot of really useful information. So one thing to really highlight is Anopheles thalabocensis, which is the main vector of Plasmodium knowlesi within Sabah, has very distinct biting patterns. So really, this vector is primarily biting outdoors, so outside of the house, but in early evening time, so between 6 p.m. and 10 p.m. So when we think about this, this is really when people are very unlikely to be sleeping inside under a bed net, and people are often out doing other activities and may be exposed to risk. We can also, by doing some of this long-term monitoring, see the monthly variations in the numbers of mosquitoes. So certainly there's variations based on rainfall and other environmental parameters, and we see some months that have much stronger seasonal peaks. We can also look at this spatially. So we've developed a number of predictive models to actually estimate the number of mosquitoes that would be biting a person outside a particular location and a particular time. And then kind of finally, of course, we wanted to look at people. So how are people moving through these landscapes? Where are they likely to be exposed to risk? And how can we collect better data on this? So this is an example again from Sabah, from a large-scale GPS tracking study we conducted to look at fine-scale human mobility and understand where people might be exposed to mosquitoes. And so it's a bit difficult to see on this picture, but this child is actually wearing this kind of small GPS tracker. I think it was initially designed for running. So this is sort of the precursor of a Fitbit or Apple Watch. But what this does is this can collect GPS data, so at a very kind of fine spatial scale, and really look at when an individual is moving through different habitat types. So from these types of data, we can collect a number of different types of parameters. So for example, we can look at individual heterogeneity. So where is a particular individual moving? Are there individuals in the community that are moving much further or spending more times in forests than other individuals? But also using all of the data and some of the spatial predictors that we've derived from these different sources of Earth observation data, we can actually develop predictive models to estimate for a given community what are the number of nights that people, or what is the number of people that are likely to be in a given location in a given time. So putting all this together really allows us to start to understand how landscape impacts zoonotic malaria risk. And so kind of just to go back, we have these detailed maps that we have of land cover. So we're monitoring the environment, where and how it changes. We also have these predictive models of human space use, so we can work out the probability that a person is likely to be outside during the times that mosquitoes are biting. Again, we also have these spatial and temporal models of mosquito biting rates. So how many mosquitoes are likely to be coming to a person at a given time. And by integrating this with the probability of mosquito being infected or infectious with Plasmodium knowlesi, we're actually able to look at where people are likely to be exposed to pathogens. So one of the kind of key things that we find from the study is really that this type of malaria is very much associated with forests and forest edges. So exposure is much higher close to the forest, but also close to the houses where people are most likely to be. So really this interface where we're seeing these different populations interact is really quite critical for driving transmission. And of course, while all of these studies have been done in kind of very detailed and very small spatial scales, we want to look at much wider population level risks. And one of the ways that we have actually done this is to run large-scale cross-sectional community-based surveys to assess exposure and infection to different malaria species. So across 4 districts in northern Sabah, Malaysia, we actually geolocated all kampongs within this region. We stratified these by forest cover, so we had a representative sampling frame of the different areas we wanted to survey. And we sampled over 12,000 people across 180 clusters or kampongs in this area. So for all of these villages, we went and we randomly selected households from each village. For the selected participants, we collected quite detailed questionnaire data on potential risk factors, such as contact with macaques, different mosquito prevention practices, access to healthcare, as well as occupation and other demographic factors. For all individuals, we collected blood samples, which we then used to look at malaria by microscopy, but also by molecular methods, much more sensitive. And we collected blood samples that we could use to assess antibodies, to look at past exposure to Plasmodium knowlesi as well as other malaria species. So as an example of kind of some of the information we got from this, using these molecular methods, we were able to identify a number of asymptomatic human infections, multiple malaria species. These within this site actually included Plasmodium cynomoldi. So this was the first documentation of this in Sabah. We also used serological assays to estimate recent exposure to Plasmodium knowlesi. So these graphs below actually show the antibody titers using the assays that we developed. This is from clinical Plasmodium knowlesi cases. So we can see kind of within the first month to year after infection, there's a very pronounced peak in these particular markers. So we're able to actually identify who is likely to have been exposed to Plasmodium knowlesi within the past year. And integrating this with all of our different data on land cover, we were able to work out associations with risk. So we found that individuals who live next to these very fragmented and quite patchy landscapes had increased risks of exposure to Plasmodium knowlesi within the past year. But we actually quite interestingly found that people that lived next to large forests that were intact and not disturbed, such as near national parks, these were actually protective. So these individuals had much lower risks of exposure to Plasmodium knowlesi. And so moving forward, the project that we're currently working on and collaborating with on is called the Sensor Project. We've spent a lot of time looking at trying to identify some of these risks and these different populations and what might be driving transmission. And really, we feel the next step to this is actually thinking about how we can use all this information about risk to design better surveillance approaches. So this is the project we're working on now, which is a 5-year project looking to develop enhanced surveillance approaches that actually think about how we can link health and environmental data to detect, monitor, and predict zoonotic disease outbreaks. So knowing what we do about forest loss and some of these factors that influence risk, how can we design better surveillance? And for this project, we're focusing again on malaria, but also looking at dengue and emerging arboviruses, such as chikungunya and Zika viruses. So these are all pathogens that can have non-human primate hosts, which are transmitted by mosquitoes. And so really kind of what we've been thinking about for this project is really how do we kind of use some of these pathways? How do we leverage technology to actually collect better data on changing landscapes? So for example, thinking about how we aggregate some of this satellite data to generate near real-time alerts, create this in a system that we could actually use on the ground, and then think about how we can use this information to design specific control measures or integrate this with health systems. So with that, I will end here, but just to kind of acknowledge the many people who have been part of this work. This is quite a large collaboration and a large research project. And thank you very much. So I don't know if there's any questions.
Thank you, Dr. Kimberly. That was very insightful presentation on your work, and it is a classic example of One Health approach in tackling malaria, I suppose. Okay, so we already have a question for you, Dr. Kimberly. So this is from Sarah Ahmad. Can the data be used to prevent and treat yet? Can we give prophylaxis or treat the macaques?
Excellent question, because that is really something we're hoping to think about how to use this. It is very challenging to control because of this zoonotic reservoir. So we've been looking more and more about what are potential options to treat the macaques, particularly thinking if there's kind of areas that have very high risk, could we deploy a targeted intervention towards macaques? So there's a lot of laboratory data that shows the effectiveness in antimalarials on macaques, mostly because they're used for testing, but we don't yet have any data to show that this is effective in the wild. And of course, one of the things we would be concerned about is whether or not we would generate drug resistance. So we're hoping to explore that and to think about how we could potentially do an intervention that was targeted at decreasing transmission within the macaque population.
Thank you, Dr. Kimberly. And we have another question from Sarah Ahmad as well. What about prophylaxis for people in forest ranges, I suppose?
Yeah, it's another really good question. And it's something that we've seen evidence of, not for zoonotic malaria, but for other malaria species within the Mekong region. So certainly, I think there's a strong case to be made that if we know that specific populations are at risk, we should be targeting prophylaxis. So for example, some of these logging camps and these very high risk populations would be really kind of ideal targets to do this. So at the moment, this isn't done routinely, but this is something we have been discussing at much wider levels.
Okay, thank you, Dr. Kimberly. So from the attendees, do we have any more questions? Maybe we still have about 5 more minutes. We can take a few more questions. Okay, I have a question for you, Dr. Kimberly. While waiting for the rest to ask, maybe I can ask first. So did your study include the migration of people from these regions where they are at high risk to a more urbanized region, and that contributing to the transmission to more urban areas of Malaysia?
We didn't specifically look at it. It's an interesting question because we know that human movement is very much associated with risk. What we tend to find is that cities and urban areas have much, much lower risk of zoonotic malaria. And I think a lot of it is driven by the mosquito distributions. So we know that there's macaques in cities, obviously, but generally the macaques which are in cities tend to have much lower infection rates of the parasite. And there doesn't seem to be as much transmission to people. So in some ways, some of the development of kind of larger cities or larger areas may actually be decreasing risk, but we still see kind of at the edges where we really see these outbreaks flaring up.
Okay. Thank you, Dr. Kimberly. So any more questions from the attendees? We have one question. Wait. Okay. So we have another question, Dr. Kimberly, from Professor Lau Yiling. So she says, thanks for a wonderful talk. Do you have any plan to expand this study to other parts of Malaysia?
You're speaking with the boss. So I think I think it would be really good to think about how we can do something. Most of our work is focused on East Malaysia, and it would be good to think about how we could do something between East and West Malaysia, because they are facing quite similar issues. But Prof. Yiling is the expert on this. So yeah, it's something that I think there's a lot of potential to do, and I think we were also seeing more countries even outside of Malaysia facing similar challenges. So Thailand's, I think the number of zoonotic malaria cases in Thailand has either doubled or tripled in the past year. So it's really increasing, and it is a regional problem. So it does need regional collaboration.
Thank you. I hope that answers your question, Prof. Lau Yiling. So we have another question here from Sarah Ahmad again. Can the government make prophylaxis compulsory for developers to give out to workers and people living there?
To be honest, I can't speak on behalf of the Malaysian government, so I don't know. I think there is a strong justification, and when we've spoken to some of our colleagues, certainly some of these industries that are very associated with risk, they do need to do environmental impact assessments, and there's a strong case to be made to say, if a logging company is opening up a new area, they should make sure their workers are protected. So I don't know what the mechanism for that would be, but I think that is something worth exploring.
Just to continue with that question, so do we have enough evidence to inform the government that this prophylaxis should be made compulsory?
We haven't seen any large-scale trials of this. We do know, so Plasmodium knowlesi is still susceptible to most antimalarial drugs, so it is quite treatable. I think it would just need to be kind of looked at more holistically to think about what are the costs and benefits, and maybe what are some of the other potential interventions. So for example, vector control and trying to limit exposure, would that be equivalent to using prophylaxis? So none of that has been looked at systematically, at least to my knowledge. So.
Okay, thank you. Question here. May I from Sarah Ahmad as well.
Good questions.
So do you have an answer for that? May I know where the name knowlesi came from?
I do know that. I think it was from John Knowles. So it was discovered, I believe in the 1920s or 30s by Knowles and Dasgupta. And of course, they named it after the British clinician.
Okay. So probably this will be the last question from Jabe Jafar. So based on the current data, do you think knowlesi malaria is still exclusively zoonotic and no evidence of human-to-human transmission going on?
Excellent question, because we've actually just completed a lot of modeling work to look at this question. So we can't say for sure that it's entirely zoonotic. There has been one example that was shown in a laboratory setting where they experimentally infected a person with knowlesi. They used this infection to infect a mosquito, and then the mosquito was then used to infect another person. So it's certainly possible that it could be non-zoonotically transmitted. From our modeling work that we've done in collaboration with the Ministry of Health in Malaysia, it does appear that the transmission in Malaysia is almost entirely zoonotic. So we don't see the spatial and temporal patterns that we would expect if we were seeing chains of human-to-human transmission. So we think that's the main driver, but it's not to rule out that it could never be non-zoonotically transmitted.
All right. I think that's about it, Dr. Kimberly. Thanks a lot for the presentation and also for answering all the questions. So thank you again on behalf of University Malaya and UMHealth. So I will pass it on to Prof. Mukri to continue with the next session.